A good morning or afternoon to you. Thank you for joining us. I'm Jan Reichert, Director of Business Intelligence at the Antibody Society. Today's webinar is one in a series designed to inform and educate our members, as well as the broader scientific community, about topics relating to antibody discovery and development. Our expert speaker, Dr. Kalyani Mondal, will provide us with antibody discovery lessons from successful LILRB1 and 2 targeted myeloid suppression. Without further ado now, I'll turn the show over to our speaker.
Thank you, Janice, for this kind introduction. Hello, everyone. Thank you for joining us for this webinar. LILRB1 and LILRB2 are two emerging targets in immuno-oncology, but they have their own challenges. So in today's webinar, I will discuss in detail how we carried out our discovery approaches at Charles River to discover drug-worthy antibodies against these two target receptors. Before I get into the specific case studies, I do have a few introductory slides about Charles River's capabilities. So Charles River is an ensemble of many sites, each of which bring their vast experience and expertise to help our clients with integrated drug discovery and development. So for example, our South San Francisco site where I work, we have a decade's worth of experience doing antibody discovery and engineering. So moving chronologically from left to right, we can support drug discovery. Post drug discovery efforts, the hits can be reformatted to antibodies. They can then be assessed for in vitro pharmacology at our sister sites at Leiden or Portishead. We also have the high throughput retrogenics platform to carry out off-target screening against thousands of recombinant proteins. And then post in vitro pharmacology characterizations, a few of those leads can be moved into in vivo disease models. Mouse-based disease models is something Charles River is very well recognized for. But not only in vivo, we also have ex vivo 2D, 3D tumor models for biological, for assessing the biological efficacy of the lead candidates. Our site at Reno has large animal capabilities, so they can help with cyno PK, PD profiling. We also have biologics division and bioanalytical services to support manufacturability and downstream characterizations. So all the way from drug discovery to IND enabling studies can be supported by Charles River under one master service agreement. So expanding a little bit more on our antibody discovery capabilities, we utilize in vitro phage display for drug discovery purposes. And the table here summarizes the different libraries that we offer our clients. So going from the left to the right, we have the COSMIC library, which is a naive scFv library. COSMIC was released in 2022. It is our largest library, has 100 billion unique sequences. And some of the key attributes include that it has fully human CDRs, has high sequence diversity, is manufacturable, and has non-immunogenicity. The second naive scFv library that we have is known as the Superhuman 2.0, which was released back in 2017. This is also quite large, has 76 billion unique sequences, similar key attributes, and up to date, we have completed over 90 projects with Superhuman 2.0. We also have a VHH library known as Tungsten, and within Tungsten, we have a camelid diagnostic library we have a camelid library that is amenable for diagnostics. And we also have a humanized VHH library where the VHH CDRs are grafted onto human family 3-23 framework for therapeutic purposes. Our humanized VHH library has been very successful in CAR platforms. One of the feedbacks we have gotten from our client is that they observe low tonal signaling with our humanized VHH in their CAR platforms. We also have a single light chain library to help with bispecific modalities. So all our libraries are royalty-free. If clone exclusivity is important to you, then Cosmic Library is the option because we do not license this library out, and therefore we have better gatekeeping on the sequences that we release to our clients. So except for Cosmic, the other three libraries are also available for licensing purposes. So up to date, we have completed some 140 de novo antibody discovery programs at our site. Within Charles River, we have carried out over 1,000 antibody characterizations in the last 2 years. We have supported IND submissions for at least 65 antibodies, and 6 antibodies from our site are in clinical trials. So as I mentioned before, that we use in vitro phage display for drug discovery purposes. But as of last year, we have now also introduced yeast display technology and incorporated it into our workflow. And this way, we leverage the power of yeast display in combination with flow cytometry to screen through millions of hits and select high affinity binders for projects where that is a primary criteria. So shown here on the slide is how yeast display complements our current phage display platform. The top figure shows our phage display based workflow. So in general, we carry out QC of our recombinant proteins before they move into phage panning. In general, if the proteins pass our QC, we are able to complete discovery campaigns in a little over 2 months, at the end of which our clients can choose up to 48 unique scFVs or 24 unique VHHs for further reformatting or testing in functional assays. Of course, they do have the option of choosing additional clones from the hundreds of binders we get for an additional fee. So our standard practice is to do 4 rounds of phage panning. So with regards to yeast display, that workflow is shown at the bottom. So after 2 rounds of panning using phage, the library generally collapses to a size that's more amenable for yeast display. All the non-binders or non-specific binders are generally removed. So post 2 rounds of phage panning, the library is transformed into the yeast vector for yeast display, and then the next 2 to 3 rounds of enrichment happens via yeast sorting using flow cytometry. Some of the selected high affinity clones then go through the single clone analysis shown at the bottom middle plot. So this is an example of a novel clone from an internal project shown in green that showed similar MFI fold shifts as the positive control, which was known to be a high affinity binder. So post single clone analysis, we do Sanger sequencing, and then the hits can be reformatted to IgGs for additional characterizations. I would now like to also discuss our retrogenics platform. Increasingly, there is acceptance within the biotech industry that such off-target assessment as well as developability assessments are best done during early drug discovery rather than during the development of lead candidates. This helps to mitigate risks upfront. So shown here at the top is a simple figure that explains how the retrogenic cell microarray technology works. So we have over 6,500 human proteins that are encoded with GFP and can be expressed in human cells. These are then allowed to bind with the lead candidate or a few of your target candidates for this high throughput screening. Binding to any expressed protein results in gain of binding function via fluorescence that is then imaged to provide a qualitative assessment. So using the retrogenics platform, we can do lead candidate specificity screening to filter out polyreactive candidates. This format has also been used for receptor or ligand deorphanization purposes, and also further deconvolution of the target itself to better understand the biological MOAs. So shown here is some internal data where we found that of the 133 antibodies that were interrogated via the retrogenics platform, at least almost half of them showed some level of off-target binding. And when we zoom into that data set for additional details, we find that 11% had polyreactivity, 16% showed binding to 2 to 3 off-targets, and 20% had binding to at least 1 off-target proteins. And on the right-hand side, I have shown here some published reports on some well-known clinical molecules that had clinical liabilities related to such polyreactivity. And so the key message here is that it is important to assess antibody specificity at a very early stage as part of the de-risking step. So shown here is some internal effort between our antibody discovery site and the retrogenics site, where we took a few of the anti-SERP alpha antibody clones for assessment against 300 frequently hit off targets. So the left-hand side figure is a representation of an array format of the data, where we took 5 anti-SERP alpha clones as SCFVs. The format design is shown above that figure, as SCFVs that were secreted into E. coli periplasmic extracts. So these were unpurified crude extracts. We included an anti-HER2 as a well-behaved negative control and assessed them against some 300 off targets, and were very happy to see that these clones were only binding to SERP family members, which is to be expected based on the panning strategy. And the anti-HER2 negative control also showed binding to its specific target. No polyreactivity with other off targets were observed. When these same clones were reformatted into antibodies, we saw broadly the same results. So once converted to antibodies due to the cross-linking via the 2 Fab arms, the assay did have improved sensitivity. So we saw binding to additional SERP family members, but in general, the clones showed binding only to SERP and not to other off targets, except for PO2-DO1, the first antibody, that showed some binding to collagen 15A protein. And therefore, we are now offering this assay to our clients where they can do this rapid assessment of up to 96 periplasmic extracts against 300 frequently hit off targets. And from this small study that we carried out, it showed that at least SCFEs in crude extracts that are outputs from our library are definitely compatible with the Retrogenics platform. So moving on to some in vitro functionality within Charles River. So we are able to support a number of functional assays, even with primary immune cells, such as activation or inhibition of enzymatic activity or signaling pathways, or apoptosis, or effector functions such as ADCC, ADCP, or CDC. So on this slide, there are cartoon representations of experimental setup and their associated data sets. We also have 2D, 3D tumor models to assess for cell killing. And at the top is the formats of these co-culture assays that are carried out. And at the bottom are examples of some data obtained with a couple of the antibodies. So up till now, I have provided a broad overview of Charles River capabilities in support of drug discovery projects. Next, I will discuss the Superhuman library. This is the library that was utilized for the LILRB1 and LILRB2 discovery campaigns. So I will go over how this library was built and what are the key attributes. So Superhuman 2.0 is a second generation naive SCFE libraries. With the first generation of naive SCFE libraries, there were many drawbacks and liabilities that are documented in the middle of the slide. So for example, if these libraries were fully natural, they had high sequence redundancy. As a result, there were very few hits and very extremely weak binding affinities. KDs were micromolar. If they were fully synthetic, then they had greater sequence diversity, but they suffered from low molecular fitness. As a result, once these clones were reformatted to IgGs, there was suboptimal folding of CDRs. As a result, they had higher propensity towards aggregation or show non-specific binding. So keeping these drawbacks in mind, the Superhuman 2.0 was built to improve on these. And this is shown in the schematic on the left-hand side. So for building Superhuman 2.0, the CDR heavy 3 and the light 3 were sourced from naive B cells from healthy human donors, and the minor CDRs were sourced from memory B cells to, so the CDR heavy 3 being sourced from naive cells was to ensure for high sequence diversity, and the minor CDRs were sourced from memory B cells to improve upon the affinities. And these CDRs were then combined and grafted onto 4x4 drug-worthy frameworks that are listed at the bottom. These are fully human germlines that were utilized, and these frameworks are well known to be manufacturable. There are clinical drugs and approved drugs that have utilized these frameworks. And so through a combination of careful framework selection, using only human CDRs, applying thermal and expression selection pressures during construction of the library was engineered for enhanced thermal stability, low immunogenicity, and low aggregation. On the right-hand side is an internal data set where we compared Superhuman 2.0 against a few published first-generation naive scFv libraries. And what is apparent visually immediately is that some of these first-generation libraries, even though they were reported to be large in size, a few clones were enriched to the extent that they took over a large percentage of the clonal real estate. On the other hand, Superhuman 2.0, which is shown in this orange dot plot, it had very low clonal redundancy. So no single clone is more than 0.03% of the total library. So the library is truly large and has very high sequence diversity. So these key attributes that I have just mentioned, how do these translate when we carry out discovery campaigns? So again, going from the data on the top left-hand panel. So first of all, across the many targets that we have interrogated with Superhuman 2.0, we generally see hundreds of unique binders that have very low percentages of high-risk liabilities, such as presence of cysteines or glycosylation sites within the CDRs. Moving on to the next top panel, in general, we see a wide range of affinities, and this speaks to the high sequence diversity that's present in the library. And sometimes that is more desirable than having just very high affinity binders. In general, there is always a subset of clones that have nanomolar affinities, and for certain targets, we have also obtained subnanomolar affinities, although I will say this is very target dependent. Moving on to the bottom left-hand panel, we generally see broad epitope coverage across whenever we carry out discovery campaigns. So shown here is one example where when we grouped our binder hits against 5 different recombinant proteins that included 3 isoforms, 1 cross-species, and 2 cross-family member binders, we could group the over 170 binders into 21 epitope bins. And then we do get developable clones. So that's represented in the bottom right-hand panel, where we took several 30 or 40 antibodies from our campaigns, converted them to IgGs, and measured TM and TAG, and they show optimal stability profile. That does seem like we have developable antibodies. It's worth reiterating that Superhuman was released back in 2017. It is the most utilized and hence also the preferred library amongst our clients, and that we have completed over 90 projects with Superhuman 2.0. We have been able to apply the library across many disease indications, so not only immuno-oncology, that does take a large fraction of the discovery programs that we have carried out, but we have also applied it against cardiometabolic disorders, against autoimmune or inflammation, again, infectious diseases, and also to discover anti-idiotypic antibodies. And moving on to the pie chart on the right-hand side, we have applied Superhuman 2.0 across challenging targets, and this includes multi-pass membrane proteins, peptide MHCs, small molecules, and even functional groups. So moving forward, the rest of this presentation will mainly be focused on the 2 case studies. And I would like to start by providing some background information about LILRB1 and LILRB2 target receptors. So immune checkpoint blockade in T cells using antibodies has demonstrated clinical efficacy in different types of cancers. So Pembro, Ipi, Tecentriq, which are all approved drugs have validated this approach. However, these drugs have only been able to help a subset of the patient populations. So beside T cells, innate immune cells such as NK cells and macrophages, they also exert a pivotal role in the recognition and elimination of malignant cells in the tumor microenvironment. So LILR family members are part of these target receptors that are expressed on these innate immune cells, and they are responsible for maintaining the homeostasis in the immune cells, and they do this by having a very fine balance between the eat me and the do not eat me signal that is shown in the figure in the middle of the slide. And so tumor cells that overexpress HLA-G, they can interact with LILRB1 and LILRB2 that are expressed on these myeloid cells. They can interact with them, resulting in suppressive myeloid signaling, and thereby triggering the do not eat me signal that helps them to escape this immune surveillance. So on the right-hand side is a schematic that shows how an anti-LILRB2 antibody can bind to LILRB2 that is expressed on macrophages, resulting in the macrophage being differentiated into M1-like phenotype that is pro-inflammatory and therefore will release cytokines. These cytokines will result in proliferation of effector T cells that can then attack and cause tumor cell death. And similar principle will also be possible with anti-LILRB1 antibodies. So on this slide, I have summarized a few of anti-LILRB1 and anti-LILRB2 antibodies that are currently in early clinical trials. So the table on the top are LILRB1 specific antibodies that are in phase 1 or phase 2 of clinical trials, either as monotherapies or in combination with other immunotherapies. And the table at the bottom summarizes a few of the anti-LILRB2 antibodies that are also in early clinical trials. So this slide provides a little bit more information on the LIR superfamily members. So the LILR family member proteins, there are about 11 of them, and they can be subdivided into 2 groups. So the subgroup A includes LILRA1, LILRA2, LILRA4 through A6. These are cell surface receptors that have ITAM activating domains, and the soluble LILRA3 protein. So all of them have extracellular IG domains, and the activating subgroup has ITAM domains for signal transductions. The subfamily B consists of LILRB1 through LILRB5. So they again have 2 to 4 IG domains on the extracellular portions and 2 to 4 ITIM domains for inhibitory signal transductions. All the inhibitory and the activating family members are expressed on a broad range of myeloid cells, which includes monocytes, macrophages, DCs, and LILRB1 is also expressed on a subset of T cells. On the right-hand side is the sequence identity matrix, that shows that these have very high homology. So targeting LILRB1 and LILRB2 poses the challenge of avoiding 10 highly homologous LILR family members that are also expressed on myeloid cells. Shown here is the cross-family binding profile of a clinical anti-LILRB1 antibody that best represents this challenge that I just discussed. So this antibody, so the data here is for the antibody reformatted to scFv. So this antibody has high affinity binding to human LILRB1. So here, this is some internal data we generated. This is the top left-hand panel. As an antibody, the KDs are actually in nanomolar range. And this antibody does cross with the cyno-LILRB1, which is the top middle panel. But it also binds to human LILRB2 and human LILRA1, A2, and A3 with a wide range of affinities. So our goal was to discover LILRB antibodies that would have better cross-reactivity profile than this clinical molecule that I just showed. So just to summarize our goal for the LILRB1 discovery campaign one more time. So our target, primary target was human LILRB1, but cyno cross-reactivity was preferable. We wanted to avoid all other LILRB and A family members. Our desired function was to target the ligand binding epitope so that it blocks HLA-G or the other classical HLAs. And the final format was going to be as an IgG with subclass G4 for use in therapeutic applications. So I will go over our workflow that we applied here. We generally like to start with a very rigorous target antigen QC, since this decides the success of our discovery campaigns. So we carried out some standard analytical QCs such as SDS phage, HPLC sizing, but we also like to assess for tag accessibility, since this is important for phage panning. And apart from ensuring that the protein is not degraded or is aggregated, we also want to ensure that the protein is well formed, well folded, has the optimal conformation. And we generally assess that by interrogating the binding of the target protein to its known receptor or known ligand. So for LILRB1, we assess the recombinant protein binding to HLA-G to ensure the protein had the appropriate conformation. So post-antigen QC, the recombinant proteins were utilized for phage panning. The outputs of the phage panning were sequenced, they were rearranged into unique clones, and then they were moved, they were then induced to be secreted in the E. coli periplasmic extracts. And then we utilized those periplasmic extracts to carry out high throughput primary screening. We used the Alpha-LISA platform for this purpose, and we assessed for binding to human and cyno-LILRB1, and we also assessed for binding to the LILRB and the LILRA off targets. So based on the data that we got from the primary screening, we took a smaller we triaged it down to a smaller number of hits that moved into secondary screening, which involved assessment of binding kinetics to the human and cyno protein, assessing ligand blocking, and confirming via FACS that the hits do bind to cell surface expressed LILRB1. So both the primary and the secondary screening was carried out with unpurified periplasmic extracts. Post-secondary screening, a small number of hits were then reformatted into IgGs. Shown here is a detail of our panning schema that was utilized for the LILRB1 campaign. So panning arm A, our goal was to find binders to the primary target, human LILRB1 extracellular domain, that do not cross to off-target LILRB2. LILRB2 has quite a high, among all family members, does have a high homology to LILRB1. So we generally carry 4 rounds of panning, and so we alter. So the first 2 rounds of panning was against the primary target. We included competition after round 2 to enrich for high affinity binders, and we introduced deselection after round 2 against the off-target LILRB2. Panning arm B, the main goal was to find human and cyno cross binders with deselection against all LILR family members. So here our panning strategy included alternating between human LILRB1 and cyno LILRB1, and after round 2, introduction of deselection against all the off-target proteins. Panning arm C, the main goal was to find ligand blockers that would also cross to the cyno LILRB1 protein with deselection against 3 of the 10 off targets, so LILRB2, LILRA1, and LILRA3, because these 3 off-target proteins have over 85% sequence homology. So for panning arm C, we actually generated the ligand binding domain. And so that was utilized for panning strategy C. And again, we alternated with the cyno protein, and we introduced the deselection with the 3 off targets. So post 4 rounds of panning, the outputs were sent out for Sanger sequencing, and we generally prefer to do a quality check at this point. Not that it guarantees for binders, but it does give us a viewpoint as to whether the panning worked. So the quality check that we do, the data is shown at the bottom. So after 4 rounds of panning, it was nice to see a wide distribution of CDR heavy 3 length and sequence diversity in CDR heavy 3. And if you look at the bottom right-hand dot plot, it showed that we were able to obtain a nice combination of enriched clones as well as singletons. So had the panning been over-enriched and collapsed, that would have been a red flag. If there was no enrichment at all, that would have been a red flag also. So we were happy to see that we saw about 20% enrichment and about 80% there was singletons. So post panning and post Sanger sequencing, the unique clones were induced into periplasmic extract, ready for the screening. So this slide expands more into our screening strategy. So looking at the left-hand side is the primary screening that we carried out. We broke our primary screening into 2 studies. The first primary screening we carried out was towards human LILRB1 full-length ECT, to the ligand binding domain, to the cyno protein, and to one of the off targets, LILRB2. In the middle is a schematic of the AlphaLISA platform. This is a bead-based proximity assay. So for example, in our workflow, we allow biotinylated antigen to bind to the SCFVs and periplasmic extracts. These SCFVs carry a specific tag. So once the antigen binds to the SCFV, the respective donor and acceptor beads come into proximity, resulting in energy transfer that then releases light that gets detected. So our first primary screening was against 4 targets, and then we had a second set of primary screening against all 9 off-target proteins. And in our workflow, we then took all the clones that were human and cyno, cross-reactive, had low to no cross-reactivity to off-targets, and then moved it into secondary screening, which is summarized on the right-hand side. So the next couple of slides will show the data that we got from our primary screening results. So this slide is a little data heavy. So let me walk you through the results that we got, starting with the correlation plot on the left-hand side. So the first plot that you're seeing is from the first primary alpha LISA screening, where we screened against 4 targets. Right. So the x-axis is the signal to noise for the off-target LILRB2, and the y-axis is the signal to noise against human LILRB protein, cynoprotein, and the ligand binding domain. And so the bottom left-hand quadrant here below the dotted lines are the non-binders. The bottom right-hand quadrant are all the LILRB2 binders. So we did see some off-target binding. And then the top left-hand quadrant are the LILRB1 binders. And so we did include the positive control, which showed binding to all 3 proteins. It's not plotted here, but the data is reported in the bottom of that correlation plot. So the positive controls and the negative controls that we included in this alpha LISA assay behaved as expected. And what we found was that there were at least 153 unique clones that showed binding to the ligand binding domain and also showed binding to full-length ECD. And there were 18 unique clones that showed human cyno cross-reactivity. So the middle correlation plot now dives into the LILRB cross-reactivity against the 672 clones that we screened. So again, the bottom left-hand panel is, bottom left-hand quadrant are the non-binders. The right-hand quadrant are the LILRB2 only binders. The top left-hand corner are the LILRB1 only binders that do not cross to or cross very weakly to a few of the LILRB family members. And the top left-hand corner are what I call polyreactive clones. They showed binding to all the LILRB family members. So the result from this middle plot is reported as a bullet point below that. So there were 194 unique clones that showed no cross-reactivity to LILRB2 to LILRB5 family members. And then if you look at the right-hand correlation plot, that evaluates all these 672 sequences against all the LILR-A family members. So the x-axis is now LILR-A1 signal to noise, and then the y-axis is binding to LILRB1 and LILR-A2 through A6 recombinant proteins. And again, the quadrants are very similar, that the top left hand are only LILRB1 binders that do not cross to any LILR-A family members, and the top right hand quadrant plots all the clones that show polyreactivity. So after the primary screening, we had 101 unique clones that showed no cross-reactivity to LILR-A and LILR-B family members from this high-throughput alpha LISA screening. And so we took the subset from this primary screening data. We took about 296 well plates worth of clones and did some additional screening and characterizations. So the left-hand side is as an isoaffinity plot that shows the binding kinetics and affinity to human LILRB1. The red dot in this plot is the 15G8 positive control. And as you can see from this isoaffinity plots, there were several clones that had improved affinity and some falling even in the nanomolar range. The middle panel shows some, as an example of some FAT screening that was carried out with periplasmic extract. And we had a good number that confirmed for binding onto cells. And of that, at least 30 clones showed MF5-fold shifts that were actually better or comparable to the positive control. And then the right-hand panel shows an octet-based ligand blocking assay that we carried out. This is some example sensograms from the larger screen that was carried out. So on the top are the experimental steps that have been reported here. So as a first step, we captured the scFv via its tag from the periplasmic extract. That is not shown in this data set. This is all processed post step 1. So once we have captured sufficient amount of scFvs, we then allow it to bind to human LILRB1, which is the first phase of these plots. And once sufficient LILRB1 binds, we then dip the sensor either into the HLA-G tetramer or a control anti-His. The LILRB1 has a His tag, so the anti-His should be able to sandwich. And so the second half is shown by traces in red or green. So green represents the sandwiching of the anti-His. So it's very easy to visualize that in the first phase, there is binding to the primary target LILRB1. And in the second phase, there is sandwiching of the anti-His. However, we have the top two panels shows also sandwiching of HLA-G tetramer, which means that these two clones are non-blocking. But we do have the other 4 clones that do not sandwich with the HLA-G tetramer, and therefore they do cause ligand blocking, which is an attribute that we are looking in our hits. So I have next 2 slides where I will show some examples of clones that do show drug-worthy attributes. So on this slide are 2 examples where they show high affinity binding to the primary target, human LILRB1. They do not cross to the cynoprotein, but they also do not cross to other cross-reactive family members. The middle panel here is confirmation via fax that they do bind to cell-expressing human LILRB1, and the right-hand side panel confirms that these clones are in the D1, D2 domain that causes the HLA-G blocking. So they do bind to the right or the desired epitope within LILRB1. And here are 3 examples where we do see cyno cross-reactivity and we see some weaker interaction with one of the 10 cross-family members. So in this case, the data has to be looked at from top to bottom. So tracing from the left-hand side, DB02A10 shows weak affinity bindings to both human and cynoprotein, but then does not cross to any of the other LILR family members. This is the one attribute that we were looking for. If you look at the middle panel, it does bind to cells. And then at the bottom panel, it does block the HLA-G tetramer. If we look at DB02C10, which is shown in the middle column, this is a high affinity clone that binds with high affinity to human and cyno, does have a 5-fold weaker affinity to LILRA3. In the middle panel, it confirmed for binding via fax, and if you look at the bottom panel, it confirms to be a ligand blocker. And then the right-hand side clone, DB02B11, it has a medium affinity to human and cyno LILRB1, does cross to human LILRA1 with a 2-fold weaker affinity. Again, confirms for binding to cell surface express LILRB1. And again, as at the bottom panel, it does confirm to be HLA-G blocking. So once we assessed the secondary screening data for LILRB1 campaign, we tried to rationalize these trends that we observed, which is that with cyno cross-reactivity, it came with some cross-family member binding. So for this purpose, here I have aligned the ligand binding domain of the cyno LILRB1 with the human LILRB1 as well as the LILRA1 and LILRA3. And the amino acids that are highlighted in yellow are different between the cyno and the human protein. So to get cyno cross-reactivity would require binding to amino acids that are not highlighted. And if you observe, those are exactly the same amino acids between the human B1 and the A1 and the A3 protein. So it's very challenging to get cyno cross-reactivity and also avoid the A1 and A3 binding. There are some amino acids here that are highlighted in green that are different between all 4 the proteins. So for example, if we look at residue number 26, which is asparagine for cyno and glycine for LILRB1, so if those are critical for human cyno binding, at least there it's an isoleucine or serine in A1 and A3. So there is some possibility of avoidance, but there are only 4 such residues. So on the right-hand side is the sequence identity matrix for just the ligand binding domain for these 4 proteins. And so what we see is that this high sequence identity, almost 80% between cyno, human LILRB1, A1, and A3, makes it challenging to ensure cyno cross-reactivity while avoiding off-target binding. If we now take away the cyno cross-reactivity, and if we only look at human LILRB1, A1, and A3 ligand binding domain, then if we look at the sequence identity matrix on the right-hand side, then that sequence identity goes up. It is now more than 85%. But now, if you look at the amino acid sequences that are highlighted in green, there are about 10 such sequences within this domain that are different between B1, A1, and A3. So even though there exists high sequence identity between these 3 proteins, there are several amino acids that are different between the 3 proteins that allows the possibility to do more specific binding to human LILRB1. But once you introduce the cyno cross-reactivity, it seems to add this challenge of completely avoiding the cross-family member binding. So to summarize the results that we got from our LILRB1 discovery campaign, first of all, the Superhuman 2.0 was successfully applied in discovery of several anti-LILRB1 clones that showed improved cross-reactivity profile than a first-generation antibody that is currently in clinic. This data set highlights the advantage of utilizing a large and diverse naive scFv library in combination with a well-designed panning strategy to identify multiple anti-LILRB1 antibodies that showed low to no cross-family binding. Also, screening hundreds of antibody hits against multiple off-targets, that can be laborious and time-consuming. And so here I have discussed the different screening methodologies that we applied at Charles River to overcome these challenges. And this included utilizing an automated high-throughput screen that assessed over 8,000 interactions within a day, with complementing this with a more rigorous cell-based functional characterizations. So moving to the second case study, which is the LILRB2 antibody discovery. So here our project goal was, again, to discover human LILRB2 binders, but here cyno-cross reactivity was more nice to have rather than preferred. Again, we wanted to avoid all the off-target proteins. Again, the desired function was to find binders to the ligand binding epitope and to be reformatted as IgGs for applications, for therapeutic applications. So this workflow is very similar to our LILRB1 workflow, so I will not go through it in details. I will only highlight some key points here. One, that during phage panning, we selected for LILRB2 and we deselected against LILRB1 close family member. And here again, we saw, if you see the bottom data set that shows the CDR heavy tree length distributions, we found, I think, over 600 or so unique binders. So nice sequence diversity post-phage panning. We screened some 7 or 8 96-well plate worth of unique clones into primary screening using alpha LISA, and the experiments, the off-targets and the primary targets are shown here as bullets, and the data set is shown again as a correlation plot at the bottom, where we looked at LILRB1 and LILRA3 as off-target binding, and LILRB2 was our primary binders. And so we got 634 unique LILRB2 only binders that did not cross to LILRA3. And since cyno-protein was not included in phage panning, we did not get any cyno-cross reactivity, although we did assess for it during our primary screening. And then from primary screening, we moved into secondary screening, which was very similar to the LILRB1 workflow, except that here all the off-target screening was carried out via fax instead of via the alpha LISA. And the isoaffinity plot is shown here at the bottom. The red dot is the positive control included. And very similar to the LILRB1 campaign, we did obtain several hits that had better kinetics and affinity profile than the positive control. So in the subsequent slides, I will show some functional data that was shared with us post reformatting these hits into IgGs. So this is some data that was shared with us by KBio, with whom we had collaborated on this project. The left-hand side represents the experimental setup. So KBio scientists, they took cells that express LILRB2, allowed it to incubate with the reformatted LILRB2 specific antibodies, and then they added or titrated HLA-G recombinant proteins to see for robust blocking. And that data set is shown on the right-hand side. They screened 15 IgGs in this experimental setup, and they observed robust HLA-G blocking, especially the clone that has an arrow on top had really good blocking of the HLA-G. They also, at their end, with the IgGs, reconfirmed the off-target binding specificity. So they expressed all the LILRA family members. So the LILRA homologs are shown on the left-hand plots. And they screened several antibodies and saw only minimal cross-reactivity to some of the LILRA family members. And the right-hand plot shows the flow-based binding of these antibodies to different LILRB homologs. And again, if you focus on the arrow, that's their KB16A41.2, their preferred clone, that basically showed no cross-reactivity to either LILRA or LILRB homologs. And then they also looked at the binding to the primary target, which is the LILRB2 expressing HEC293 cells. They saw a wide range of MFI fold shifts, which represents a wide range of affinities. And again, their lead candidate had about 121-fold shift over background, which shows that it is likely a very high affinity antibody. So I have some additional functional assay data with their lead antibodies. So this, the slide here really goes into some details of the functional assay setup. So LILRB2 expressing human monocytes, they produce pro-inflammatory cytokines such as TNF-alpha when exposed to stimulus such as LPS. And if these primary immune cells are co-cultured with HLA-G expressing tumor cells, then the HLA-G would interact with the LILRB2 that is expressed, as shown in figure 2 here, and would result in suppressive signaling that would then cause a reduction in the cytokine production. And now if an anti-LILRB2 is added to this co-culture that then disrupts this tumor cell engagement, then there is rescue of the cytokine production. So the same schematic that I showed in the previous slide is shown here again. So in this case, K-Bio, they carried out some co-culture assays with stimulated monocytes that they co-cultured with JEC3 cell line. This is a carcinoma cell line, and then they monitored the levels of TNF-alpha. And so that data set is plotted on the right-hand side, where they took macrophages that have been stimulated, and upon stimulation, they have a reference of the TNF-alpha levels. Once they add the JEC3 cells, there is a substantial drop in the TNF-alpha level shown by the gray column. And when they titrate their anti-LILRB2 antibody, they start to see the rescue of the TNF-alpha levels. So an antibody derived from our libraries shows functional activity in some of these co-culture assays.
Kalyani, we're already 5 minutes over.
Okay, I will then quickly summarize. Thank you for that. So then, just very briefly, this is some additional functional assay they carried out, where they assess the macrophage phenotype. And so they do see upon addition of the antibody to the undifferentiated macrophage, it becoming more pro-inflammatory, resulting in more TNF alpha, and the anti-inflammatory signal, which is monitored by IL-10, actually goes down. They are carrying out some in vivo studies with xenograft mouse models. And so to quickly summarize, I will, in consideration of the time, I'll not go through all the bullets here. I'll just go over what are the next steps within these two programs. So for anti-LILRB2, this is from KBio, that they're carrying out additional in vivo studies in monotherapy as well as in combination. And next steps for anti-LILRB1 is that there is a possibility of additional engineering, either to improve the clone that had human and cyno cross-reactivity, or for clones that had some binding to the off-target that could be maybe reduced. And I think for the audience, the main takeaway is that the lessons learned here can be applied to next generation immune checkpoint inhibitors that can require recognition of multiple isoforms or require avoidance of multiple close family members. An example of this is SIRP alpha, which is also one of the immune checkpoint inhibitors that are being looked at. So with that, I do want to acknowledge our partners that allowed us to present this work. I want to thank Antibody Society and Janice for allowing me to present this work on the Antibody Society platform. I want to thank Charles River and our site head, Rennie Feldman, for their support. And our team, here's a picture on the right-hand side. I want to thank the team for their important contributions for these two programs. I also want to thank the audience for their time, and please feel free to reach out to us for any questions around your specific projects via our website. So with that, I will stop and happy to take any questions.
We do have one quick one, and that is, what is the false positive rate of the retrogenics off-target panel by your experience over time?
I think this generally varies. So from our libraries, we have seen very little off-target binding. In general, it's not been more than one, but it can vary. It can be very target dependent also.
Now, one more quick question. Why did you choose IgG4 as the isotype?
I think this is it came more from our partners, and I think it's likely due to the clinical molecules. They have IgG4, and therefore they probably opted for this. IgG4 is also quite low on effector functions, so you get more any kind of immune readout is very target specific rather than coming from any effector function. There are many approved drugs within immuno-oncology or even outside that use IgG4, so it's very well used and well behaved, and I think that's why our partners chose the IgG4 subclass. But it's possible that you can have an engineered IgG1 to be effectorless, and that may also work well.
All right. In 2 words, respond to the question, are the libraries for sale or only for service?
We do out-license, yes.
Okay. Thank you so much.
Thank you so much.
I'd like to thank Dr. Mondal for providing antibody discovery lessons based on her expertise and knowledge of CRL's phage display libraries. And for the audience, the PDF is available, and you can certainly feel free to reach out to Dr. Mondal with any additional questions. Thank you very much for attending the webinar today. An on-demand version will be available very soon. I'll send a link to it via email to everybody who registered. So please do feel free to watch this or any of our on-demand webinars when it's convenient. Thanks again and have a great rest of your day.
Thank you.